In section 12.8, we begin our discussion of blood vessels. We see the different types of blood vessels and their variations in anatomy depicted here on the left-hand side of our screen. What we immediately notice is that the arteries have thicker walls composed of connective tissue, smooth muscle tissue, a number of layers of elastic tissue, and veins have somewhat thinner walls, and then we progress to thinner and thinner walls as we go from venules, which we'll talk about soon, to arterioles, and then all the way down to capillaries. What all the blood vessels share in common is a single layered lining of endothelial cells that exist on the innermost portion of the blood vessel. In the case of capillaries, that's all we've got, just a single layer of endothelial cells. We see the pressure differences that we have in various blood vessels depicted here in the diagram at the lower right-hand side of our screen. The pressure is, of course, highest in arteries, and it's much higher in systolic, as you know, and as we have previously discussed. It's much higher in systemic versus pulmonary circulation. But then as we go to arterioles in the two respective systems, we have a drop in pressure. Capillaries have a much lower pressure. Venules, a much lower pressure still. And then veins have the lowest pressure of all of the blood vessels. And we'll talk about each of these in more detail as we progress here. So we begin with a discussion of arteries. Arteries have these thick elastic walls, and the purpose of arteries is twofold. They serve as low resistance tubes that transport blood from one part of the body to another, and they serve as these pressure reservoirs. So to understand how they function as pressure reservoirs, we need to understand compliance. Compliance is how distendable a chamber is. Compliance can be calculated as the change in volume over the change in pressure. The greater the compliance, the more easily structure can be stretched. So a balloon that has high compliance, if you blow into it just a little bit, then the volume changes dramatically as a result of the addition of that gas. A balloon that has very low compliance, you blow into it and it doesn't change much at all. You have to blow into it very, very hard to get it to inflate. So arterial compliance contributes to blood pressure. The arteries stretch, but then they snap back to their original shape. They have this intermediate compliance that allows them to stretch, but they are not so compliant that they won't push back to their original shape. What we notice in our figure that we have on the lower right-hand side of the diagram is this. As the ventricle contracts during systole, blood is pushed into the arteries. But what we see is this, that only a third of the stroke volume, the blood that entered the arteries, leaves during systole. The remainder of that blood exits during diastole. So the end result of this is that when the ventricle contracts, a third of that stroke volume is leaving here, but 2/3 remains in the arteries. And so the arteries stretch, the semilunar valves close, and then the arteries snap back to their original shape, pushing the blood through the body. And so in this manner, the arteries are a pressure reservoir. The original pressure is, of course, generated by the contraction of the ventricles, but it's maintained and transferred through the circulatory system by the compliance of the arteries. If we look at the pressures that we see in arteries, then we notice a couple different pressures that we want to be familiar with. The pressure is, of course, highest during systole, which is ventricular contraction. Pressure is lowest, as you would imagine, during diastole. And the difference between the systolic pressure and the diastolic pressure is known as the pulse pressure. And so the farther apart these two numbers are, the greater the pulse pressure. We also notice this, the mean arterial pressure, MAP, is not simply halfway between the systolic and the diastolic pressure, but rather it's calculated as the diastolic pressure plus 1/3 of the pulse pressure. This is because more time is spent in diastole than systole, and so to calculate the mean pressure at all times in the arteries, you have to take into account that fact that diastole is longer than systole. Mean arterial pressure is a useful number because it describes the pressure that's driving the blood through the circulatory system all of the time, taking into account both contraction and relaxation of the heart. Lastly, in this little section, we talk about the measurement of arterial pressure using a sphygmomanometer. And so this, of course, is simply taking your blood pressure. And what we notice is that, as most of us are aware, it's done in the following manner. So if we begin on the left-hand side of our diagram, then what we have done is wrap our sphygmomanometer around our arm. We've placed our stethoscope over the brachial artery, and we have inflated the sphygmomanometer until we have completely occluded that artery. And so there's no blood flowing through it at all. And so we don't hear anything in the stethoscope at this time. We then slowly release the pressure, watching carefully on the gauge, and we release it, and listening carefully as well. We release it until we hear these sounds, these sort of gurgling sounds that are made as the pressure in the sphygmomanometer drops just below the pressure that's required to occlude that artery. And when the artery is open just a little bit, blood shoots through it as a result of the pressure difference between the two sides of the sphygmomanometer there. And these sounds, referred to as Korsakoff sounds, can be heard. And so that, of course, is our systolic pressure. And then we continue to lower it and lower it and lower it. And eventually, we will lower it down to the point at which we are not occluding the artery at all. And so the sounds will go away. And so we record our second value, our diastolic pressure, at the point at which we have decreased the pressure to the extent that we're no longer interfering with the artery in any way, and we don't hear any sounds at all. And so that gives us, of course, our systolic pressure and our diastolic pressure. In section 12.9, we'll discuss arterioles. And what we notice is this, that arterioles are the blood vessels of the body that are located between arteries and capillaries. And arterioles are responsible for determining two aspects of cardiovascular physiology. Number one, they're responsible for determining the blood flow to various organs in the body. And number two, they're responsible for determining the blood pressure in the entire body, or at least largely responsible for that. The reason for this is that they have a tremendous ability to either vasodilate or vasoconstrict, depending on the needs that a various organ has at any given time. And then collectively, if you examine all the arterioles in the body, the vasodilation or vasoconstriction in these arterioles determines the overall blood pressure in the body. Now, we can illustrate how this works by looking at the little illustration below on the bottom right-hand side of our screen. So we have a pressure reservoir that is located above these various tubes, and the tubes are either constricted or not to various diameters. And what we notice is that the pressure that's pushing into each of these tubes is exactly the same, but the outflow diameters are different. And furthermore, if we look at the amount of flow in any given tube, then we notice it's directly correlated to the diameter of the outflow. For instance, in number 1, we get very little flow. Number 2, we get a lot of flow. 3, almost no flow because it's so constricted. And then the same thing for 4 and 5. And then B simply illustrates this. We can change the amount of fluid that we're putting in any of these tubes very simply by changing the diameter of the tube that goes to the particular little measuring thing below. And so, for instance, in number 2, we had a broad diameter here, lots of flow through number 2, and then in part B, we constrict that diameter and we greatly reduce the flow through number 2 in part B. That's exactly analogous to what's happening in our body with our arterioles. So the overall blood pressure that's created by the contraction of the ventricles, the mean arterial pressure, represents this pressure reservoir that we have up here at the top. And then the little tubes, of course, are analogous to our various arterioles, and those arterioles can dilate or constrict at any given time, allowing more or less blood to flow to a particular organ. Now, we turn our attention now towards arteriole diameter and what controls arteriole diameter. So if we look at an arteriole found here in the bottom left-hand side of our slide, then we see it's surrounded by a great deal of smooth muscle. And this smooth muscle is always contracted to some extent, and we refer to that as intrinsic tone. So the question becomes this: how do we either increase that intrinsic tone or decrease it? If we increase the intrinsic tone, then of course we're constricting the blood vessel. If we decrease the intrinsic tone, then we're causing dilation of that particular arteriole. And there are two ways that we accomplish this increase or decrease of intrinsic tone. One way is local control. A second way is extrinsic control. We now want to spend some time talking about the factors that control vasoconstriction and vasodilation in arterioles. We said we divide these factors up into two categories: local controls and extrinsic controls. We'll talk first about local controls, and we have three different local controls that determine vasoconstriction and vasodilation in arteriolar smooth muscle. So the first one that we'll talk about is referred to as active hyperemia. And essentially, active hyperemia is defined as this: when we have an increase in demand in a tissue, then we have vasodilation in the arterioles and an increased blood flow to that area. This response is, of course, highly adaptive, and it's best observed in skeletal muscle, as you might imagine, cardiac muscle, and glands. These are the three types of tissues in which we have the greatest variation in metabolic needs over time, and therefore it would be most effective to have active hyperemia be very functional in these particular tissues. So the basic idea is this: if we look down at the bottom at our little diagram, we see that we begin here, and we have an increased metabolic activity. We have increased metabolic activity in the organ. As a result of that, then two things are taking place. Oxygen levels are going down, and the number of metabolites in the interstitial fluid are going up. And so these metabolites include things like carbon dioxide, hydrogen ions, adenosine as the result of ATP expenditure, nitric oxide, which is a gas signaling factor released by endothelial cells, and other factors as well. So the basic idea is that oxygen's going down and all these things are going up. That leads the smooth muscles to relax, and so we have arteriolar dilation, and we have increased blood flow to the organ. The second of the local controls is known as flow autoregulation. Flow autoregulation is a form of homeostasis that counteracts flow changes and therefore keeps roughly the amount of blood flowing to a particular organ constant at any given time. So the basic idea is illustrated in our little diagram here. We have decreased arterial pressure going to that organ. We have decreased blood flow to that organ. And so just the opposite occurs relative to what we just talked about. We have a decrease in oxygen, of course, instead of an increase. We have an increase in the various metabolites that we talked about as opposed to a decrease. And then we have a decrease in vessel wall stretch in the arterioles going to that particular organ. So that means that we have arteriolar dilation in the arterioles that are letting blood go to that organ, and a restoration of blood flow from the low levels back to normal levels in that particular organ. So this can occur in either way. If we have decreased blood flow to an organ, which is what we just talked about, then we have dilation of the arterioles to restore that blood flow. But if we have increased blood flow, we have the opposite. All these arrows would just be flipped here. And so we have constriction of arterioles as a result of that, and which limits the flow of blood to that particular organ. So this is super adaptive because it roughly keeps the amount of blood going to a particular organ constant at any given point in time. Now, I hope this is clear. This differs from active hyperemia in that in active hyperemia we had an increased need of the tissues. There was no difference in the diameter of the amount of flow originally going to the organ, but we have a higher metabolic rate in the organ and therefore more flow. Here, we have either more blood or less blood that's going to the organ, and the arterioles are constricting or dilating to counteract that and keep the blood flow constant. The only thing we didn't talk about, so let's go over the mechanisms. And what we notice is this, that it's not surprising the mechanisms that are responsible for this, at least as far as oxygen and metabolites go, are the exact same mechanisms that we talked about in active hyperemia. So although the cause of that decrease in oxygen, for instance, and increase in metabolites is different, the response of the blood vessel is exactly the same as we observed in active hyperemia. Now, one thing we haven't talked about is this, that the smooth muscle in the arterioles has something of a myogenic response. This means that when less pressure is applied to it, then it dilates. When more pressure is applied to it, then it constricts. So this is what we refer to as the myogenic response. This is a result of the alterations in calcium that are available to the smooth muscle cell, and that's changed by the velocity of the blood that's passing those smooth muscle cells. And so the myogenic response is not entirely responsible for flow autoregulation, but it does contribute to flow autoregulation. The last type of local control we want to talk about is referred to as reactive hyperemia. And so reactive hyperemia is essentially an extreme form of autoregulation. And the basic idea is that if blood flow is occluded or blocked to a particular tissue for an extended period of time, and by extended period we mean a matter of minutes, then when blood flow is restored, there will be an extreme deficit of oxygen and an extreme abundance of these various metabolites. And so blood races into that area as a result of pronounced dilation of those arterioles, restoring oxygen and reducing the number of metabolites in that particular area. And so, you can demonstrate this, although I don't know if I would, by tying a string around my finger, leaving it there at moderate tightness for a minute perhaps, removing it, and then your finger will turn red as a result of the blood rushing in and restoring the oxygen concentrations in that particular, the tissues found in your finger. We now turn our attention to extrinsic controls that modulate vasoconstriction and vasodilations in arterioles. We began with a discussion of the autonomic nervous system, and in the autonomic nervous system, it's primarily the sympathetic division that's concerned with or involved with regulating the contraction state of smooth muscle in arterioles. And in this particular case, oftentimes these sympathetic neurons are concerned not just with the fight or flight response, but rather with maintaining homeostasis in the entire body. So for instance, regulating blood pressure or regulating body temperature are two roles that the sympathetic neurons that end on these arterioles play. Arterioles do have many connections with sympathetic postganglionic neurons, and they possess receptors, of course, for norepinephrine, but these receptors are alpha adrenergic receptors as opposed to the beta adrenergic receptors that we saw in the case of the heart. This means that different antagonists can be used to regulate either heartbeat or blood pressure, because although the receptors in these two locations, the systemic arterioles and the SA node, or the atria more broadly, bind to the same ligand, they're different receptor types, and so we can use different antagonists. We can synthesize different antagonists that bind to one receptor but not the other. We also noticed that these sympathetic neurons are rarely completely dormant, but typically discharge at some rate, and that rate can either be increased or decreased depending on the whole body needs, for instance, in the regulation of blood pressure. Or take temperature as an example. And so what we see is that binding to these alpha adrenergic receptors always causes vasoconstriction. And so following exposure to cold, then we have an increase in sympathetic stimulation to the arterioles that deliver blood to the skin. We have less blood flow to the skin, more of the blood is kept towards the core of the body, and therefore the overall body temperature is raised in response to that stimulation. What we also noticed is that the discussion of parasympathetic neurons in controlling arteriole or smooth muscle contraction is fairly short, because parasympathetic neurons have little or no input to these blood vessels. And this is unusual, of course. Most of the time in the body, we observe dual input from the sympathetic and parasympathetic nervous system, and the opposing forces of these two systems determine the overall state of the organ, but that's not the case in this particular instance. And so we only have sympathetic stimulation, and the overall state of contraction is determined by the degree of only sympathetic input. Lastly, what we see is this. We have some neurons that are not cholinergic and not adrenergic. They don't release acetylcholine, they don't release norepinephrine, but they do play a role in regulating arteriolar smooth muscle contraction in the body. And so these neurons are primarily found in the digestive system. They're sometimes referred to as the enteric nervous system and kind of treated as a third branch of the autonomic nervous system beyond sympathetic and parasympathetic. And so these neurons, of course, are not releasing the two common neurotransmitters, but they're releasing other neurotransmitters that typically lead to vasodilation. In fact, in most cases, they're releasing nitric oxide, pictured here on our screen, which leads to vasodilation as a result of that release. The second type of extrinsic control that we want to discuss are hormones. There are several different hormones that impact arteriolar smooth muscle contraction. The first on the list is epinephrine. Epinephrine binds to two receptors, interestingly, in the body, having an impact on smooth muscle contraction via these two different receptors. The first are alpha adrenergic receptors. These alpha adrenergic receptors lead to vasoconstriction. These are the receptors that we just talked about that are involved in the sympathetic division of the autonomic nervous system, and that these alpha adrenergic receptors also bind to norepinephrine. So there is a second type of receptor. Let's go ahead and talk about that, a beta-2 adrenergic receptor that binds to epinephrine. Interestingly, this has the opposite impact as when epinephrine binds to alpha adrenergic receptors. This causes vasodilations. Alpha adrenergic receptors, vasoconstriction, beta-2 adrenergic receptors, vasodilation. Now, this is adaptive for the following reason. So in a fight or flight response, you need less blood flow headed towards your, for instance, digestive system. You need more blood flow headed to your skeletal muscles. And consequently, what we notice is that we have large numbers of beta-2 adrenergic receptors found in skeletal muscle beds, and so the release of epinephrine goes to those and sends more blood to the skeletal muscles. Whereas, if we look back up at this top bullet point, we see that the alpha adrenergic receptors greatly outnumber the beta-2 adrenergic receptors in smooth muscle, such as the smooth muscle found in the arterioles that heads towards the digestive system. So we get less blood flow to the digestive system, more to skeletal muscle, as a result of the release of the same hormone. We can see that in our little diagram in the lower right-hand side of our screen. Actually, we're combining both epinephrine and norepinephrine and epinephrine into this diagram. So if we release norepinephrine, then it goes up in the extracellular fluid, binds to the alpha adrenergic receptors, causing vasoconstriction and less blood flow. However, if we release epinephrine, not norepinephrine, then it also binds to alpha receptors, causing vasoconstriction, but binds to beta-2 receptors in skeletal muscles and causes vasodilation. We see a couple other hormones that play a role in arteriolar smooth muscle contraction. Angiotensin II, released in the kidneys and elsewhere, and vasopressin, released, of course, by the posterior pituitary. Both cause a constriction of arterioles, and this is adaptive because it would help preserve blood pressure following event of blood loss. We also see that atrial natriuretic peptide, which is secreted, of course, by the cardiac atria, is a potent vasodilator, although its overall role in human physiology is not clear. Lastly, we want to talk briefly about secretions of the endothelial cells and arterioles themselves that have an impact on the contraction or relaxation of smooth muscle found in the arterioles. So these endothelial cells secrete substances that are typically paracrine acting, locally acting. They may secrete them all the time, or they may regulate these in response to external signals or stimuli. The first and most important would be nitric oxide. Nitric oxide is continually released and is a potent vasodilator, as we've mentioned previously. Now, nitric oxide also acts as a neurotransmitter, as we've discussed earlier in our class, but this is not the same nitric oxide, of course. This is being released from the endothelial cells themselves, having an impact on the smooth muscle in the endothelium, or rather in the arterial cells themselves, and so it's not, of course, a neurotransmitter. Nitric oxide release can be modified, for instance, in an allergic reaction. If you were stung by the yellow jacket on the screen there, then the histamine release would increase and nitric oxide would be released at higher levels, and that would contribute to the swelling that's observed. Prostacyclin is a potent vasodilator as well. It's not released constitutively, it's not released all the time, but it is released in certain scenarios, and we'll talk about its role in blood clotting a bit later in our class. And then endothelin-1, interestingly, is a very potent vasoconstrictor that's released by the endothelial cells, whereas the previous two were, of course, vasodilators. So we have a summary diagram there, figure 12.36, that looks at all the different factors that we have examined controlling the contraction state of smooth muscle in arterioles. We talked about all the local controls that are involved in active hyperemia and flow autoregulation. We talked about hormonal controls that have an impact on the smooth muscle contracting state, including epinephrine, angiotensin, vasopressin, and then some of the less important hormones like atrial natriuretic peptide. And then finally, we talked about neural controls, hormones, or rather neurotransmitters released by the sympathetic nervous system, but not parasympathetic, that modulate the contraction state of smooth muscle associated with arterioles. And so we said that the sympathetic nerves release norepinephrine, and there is no release from parasympathetic nerves, but there is, of course, nitric oxide that's released by that so-called enteric nervous system, causing vasodilation of the arterioles that make their way to, for instance, the large and small intestine.